What makes biological systems different from physical systems? In other words, what makes biology different from physics? That's an important question because we want to address how to make models of biological entities. And we might think that in the end, everything is physics and chemistry, so that's all we need to understand biological complexity, but that's not the case, right? Physicist John Hopfield you'll hear about him a lot, suggested the idea that actually what makes biology different from physics is the fact that biological systems perform computations. In other words, if I have, for example, a cell is a system that moves in a given environment, needs to adapt to that environment, needs to take resources, and needs to react. So in other words, you need to gather information from the surroundings, you need to process the information, and you have to respond. That's something that is not in the physical, non-living systems. Our first approach to biological complexity in this lecture has to do with the simplest level in a way, right? That doesn't require to look at the complexity of the individuals, okay? I just put two simple drawings here. One for a cell, right? And the cell in a very sketchy way, you could say that it's made by a compartment, right? A membrane that is more or less complex, by some information, okay, that is stored in the DNA, but not only the DNA. And inside the cell also we'll have a number of reactions that build the metabolism, what you need to actually process the matter and energy that you need to live, and do something that is absolutely essential, and that's what connects with the first part of the lecture, growth, right? Biological systems and cells in particular have a very clear distinction from the inanimate matter, which is they can replicate, they can self-reproduce, okay? So we want to see how we can model that at a given level, which is the population level, right? What things are not going to be introduced here? Well, we are not going to take into account the fact that populations are diverse, that except in the lab, we don't have single species populations. We will ignore behavior, for example, right? Behavior that can be built, for example, by the fact that you can move, as simple as that, right? But there's a crucial innovation in evolution, right? You move and you can actually search in space, right? We are not going to include that. And we can also think in different systems that for a number of reasons also self-replicate, but self-replicate at the cost of using something else, which is the case of viruses, where you have also something you can call a compartment, something that protects information. You do have information, but the vast majority of viruses are limited to that. They have to use cells to self-replicate. That's a story that we'll explore separately here, all right? So growth and death. Death might seem more boring stuff, but it is not. And we'll see, not in this lecture, but we'll see why this is relevant, okay? So what do we observe when we see a population, right? And how can we measure that? Well, this is a very classic kind of experiments. You put a given number of cells within a tank, right, with some resources. You observe on time, for example, it could be days. X is going to be my population, right? And I measure from time to time how many individuals do I have, starting from an initial amount, which is going to be called initial condition, right? It's going to be my X at time 0, right? That's my time equals 0 origin, okay? So a typical observation, for example, for some unicellular organisms, is that essentially you see very rapid growth, okay? This is sampled in intervals, right? For example, 1 day or 1 week or hours or whatever. But then this is followed by saturation, okay? So if you want, I can actually draw a line. We don't know what line is this exactly, but essentially it's a rapid growth followed by saturation. Why saturation? That's a very intuitive idea. Resources are limited, so you cannot grow beyond any limit. Okay? Whereas at the beginning, the situation is quite different. You have a lot of resources, you can grow quickly. Okay? So what is the simplest model we can formulate for growth? And you'll see that this model is extremely simple, but it reveals important things. Let's imagine that we are in a phase where resources are not limited. You can grow as much as you want. Okay? In that case, you will observe that in a number of situations, for example, in patients or model organisms with leukemia, some kinds of leukemia, you see a very rapid growth in the population of cancer cells, right? All right, this is usually called exponential growth, and we'll see where this term comes from. And it has a lot of consequences, right? From both the growth of populations, the growth of tumors, but also in the rapid spread of epidemics, okay? All right, so how can I make a model of this? Well, I know that individuals self-replicate, okay? Meaning that once I have stored enough resources taken from the environment, I grow and grow and grow, and by mechanisms that are quite sophisticated, I can reproduce, I can replicate, right? In the simplest case scenario, right? For unicellular organisms. So how can I write down this, right? Imagine that this happens with some efficiency, a rate, R, right? Which tells how efficient is the mechanism of replication, okay? In general, that would be something that weights the number of offspring that you leave in the system, all right? So it is easy to construct a mathematical model in the following way. This is going to be my population at any time, okay? This, of course, will be something between a given number of limits, but in principle, we can open this way, right? And imagine that I want to know, after a while, right? So I take a time here, for example, or here. This is my time t, anytime. And I want to know after a given interval, so this will be t plus delta t, I want to know how much the population has been growing, all right? Well, it's easy to do. Let's just follow the following steps. This is going to be something that for sure has to take into account what is already there, right? At time t, we had a number of individuals, right? Let's suppose the mortality here is not an issue, and they have been reproducing. So I have the ones that already had at this time, and now, how many extra individuals I'm putting in the system, okay? Well, this has to be proportional to the efficiency of the process, so how quickly I replicate, right? How many offspring I put in the system. On the other hand, that's a very important part of the story. The more individuals I had, the more can replicate. Okay, so it has to be proportional to that quantity. All right? Very good. And there's a missing part that we haven't yet put here, and involves the experimental approach, the fact that we take measures at finite time steps. So the larger this delta t, the more time I give to the system to introduce more individuals. So I have to also consider that as an arbitrary time scale that I use experimental, okay? So that will be the discrete equation that I will use to explain how populations grow, supposing, and the hypothesis that there are no limitations, no resource limitations of any kind, okay? All right, now let's manipulate that a little bit. Some of you will be familiar with this because it's basic calculus. I can move this at this side, right? Once I put this in this side, I can divide this by delta t, and I have this expression. What is this here in the left-hand side, right? This is the speed of growth, how quickly things are happening. Under a given time window, how fast populations have been changing, okay? It's the velocity of change. This, of course, is arbitrary because delta t is arbitrary. Delta t could be very large, could be very small. Right? We don't want that. We want something that's mathematically general. So how to do that? We can take the limit. The more small delta t is, the more precise is our fine-grained analysis of the system, right, of all this here. And that is, and many of you know, okay, this here is just a definition of the derivative, okay? So this is the derivative of x, population over time. So how quickly in the limit populations grow, all right? And this is equal to what? Well, this is r xt. From now on, we will assume that our variables depend on time. We are not going to write down this usually when we write our differential equations, right? That's the differential equation that introduces a variable x that I want to know. I want to determine who is x of t, right? What's the exact form of the curve that I have here. And this tells me what controls the speed of change in the system, right? Here it's just saying it's proportional to the number of individuals, okay? We are going to use some notation to identify this kind of mechanisms, and for this replication system that just has individuals that can replicate with no other constraints, right? We'll use this diagram. Okay? So far, so good. Okay, so now this is fine. This is an equation that tells me how the speed changes over time, but I want x. I want the solution. And for this system, things are easy to do because this system is a kind of differential equation, the simplest one you can formulate, almost the simplest one, right? You can separate the variables, put the x here, dt here, so have x and time separated, and you can integrate. Integrating between what and what? Well, at time 0, my population was x of 0. At time t, an arbitrary time in the future, I have x of t. That's what I want to find out. That's my target. Okay? Very well. So this is very simple to solve. This is a logarithmic integral. Usually we use, instead of x of 0, we use this kind of symbol. This is just r, constant that goes out, and this is t. Very easy. We are almost there. We take exponentials on both sides, and then we put x of t again. The solution of that is what's called the exponential function, the exponential growth equation. All right? That's the way a population of individuals that can replicate with some efficiency, with a rate r, grow in time, assuming that there's no limits at all in their capacity for replication, right? There's no limitation in space, no limitation in resources. So what's the implications of this, and how do we deal with that? And why is it important, right? Because you can actually think, well, yes, but this is an equation that says that if I wait, I go growing and growing and growing to infinity, right? And it's true. What I just found out here, right? Suppose this is a given R1. If R1 was smaller, I will increase at a slower rate, right? But essentially they go to infinity, okay? All right. So what can I do with that? And why is this relevant at all? Because I know that in the real world there are limitations, okay? Well, the thing is that exponential growth is not valid on the long time limit, of course, but it's perfectly valid in this domain here, okay? Where effectively cells are not experiencing any kind of limits. Okay? Very good. So what can we do with that? And why is this going to help us to understand some of the fundamental biology behind? And the trick is very simple. From these things that can be used to adjust experimental data, I can extract very, very valuable information, right? One way of doing that makes it nicer to calculate is to make a small transformation. Okay? If I take this equation and I take logarithms in both sides, the logarithm of x here will be the logarithm of this product, which is the sum of logarithms. So one constant here, right? Plus the logarithm of this term here. But the logarithm and the exponential, this is, it should be written this way. But anyway, it's a logarithm of e basis. Are inverse functions, so essentially taking the logarithm of this is doing nothing. Okay? So what's this? If I write down here logarithm of x, my new vertical axis, right? This is a constant here. Okay? This is constant here. And this is a straight line, right? So I transform this shape in a straight line. What is the slope of this line? And that's the crucial part of the story. The slope of this line is the growth rate, how fast populations grow. Okay? Which is a crucial part of the information I want. Imagine that this is a cancer cell population and I want to know how fast cancer cells grow. Or maybe I have been using a treatment and my cancer cells grow in a slower way. I need to know what's the growth rate. And that is directly determined from here. I don't need to know all the information about how is this going to saturate. I can know for sure and well, right, a crucial part of the story. Okay? Very good. Now, this is something that I can construct, and one nice property that I can derive from that is a single number that captures a lot of the information that I have here, which is the doubling time, right? What is doubling time? Well, imagine that I'm following this population here, okay? And I want to know how long I need to wait to have a double population. So twice the initial population I had in my cell culture or in my population, all right? To do that, I just need to use this equation, okay? The solution of the differential equation that I just derived from exponential growth. Since this equation is valid for any initial condition I can formulate, for any r, etc., I can take this equation, and what I'm saying is, I want to know the time t2, well, the doubling time, right? Which is defined in which way? That's the time where this is 2 times x0. All right? I replace this t by this particular t when x is going to be twice this value, right? The initial condition disappears, which is very convenient. And essentially, what I have is this 2 equals e to the r t2. If I take logarithms, now put the period logarithm, whoops. I take this, and I have the definition of the doubling time, which is in a single number, right? I capture a lot of information about how the population, how quick the population replicates. In a single number, I have I can weight how that translates in terms of the total population, right? Okay, so that's for the exponential function. Important lessons that we need to learn. The first is that an extremely simple model that clearly ignores a lot of details, right? That clearly ignores the long-term behavior it's helpful. So that's going to be helpful not only here, but in many other different contexts. We can measure things properly. Okay? So that's for the exponential growth. And now the question also is, of course, living systems not only grow, they also die. Okay? So how can I formulate a model that in a way captures that totally different idea, right? That individuals die, okay? And is death important? It's just a simple, trivial mechanism? It's not, right? Remember what Mary Shelley said. Mary Shelley wrote Frankenstein in 1818 when she was 18 years, right? And at some point in the book, very, very targeting these kind of things, Victor Frankenstein says, in order to understand life, we first need to understand death. So let's go now for the death part of the story. We have seen how to make the simplest model of population growth, assuming that individuals are extremely simple entities and that we just want them to replicate at a given rate with no particular limitations. Now, the other side of the story, of course, that we have to take into account is death. Any single individual has to die, maybe with some exception that we will discuss later on in some other lecture about aging. But in general, we have to look at individuals as objects that sooner or later decay and disappear. Again, we are going to talk about death in the simplest scale at the population level, where individuals, again, are considered entities with no particular internal structure, although we know they do have a structure. And this is relevant because structure means that you have a lifespan. Over that lifespan, you experience fluctuations and changes and deterioration that affect your internal state. And also, we have, again, at a smaller scale, a number of processes that are interacting at the gene interaction, at the metabolic network interaction scales, where changes have to do with the processes of aging. Okay? And of course, the open question for bioengineering is to what extent aging is a phenomenon that we can actually just see happening, or we can interfere and maybe revert, right? A lot of what is here has to do with evolution, right? We are not yet putting evolution in place, but the origins of death have to be also situated in the evolutionary context, as we will see. Okay? I also wanted to mention that death is a process that happens at multiple scales, also within organisms. In our own organisms, death is a constantly happening process, right? And part of this process has to do with decay. Part of this process is deeply integrated with the structure of tissues. And part of this process is due with some amazing novelty that emerged in evolution, which is called the programmed cell death and apoptosis. All those mechanisms that are active and that allow to kill cells when they might be a threat for the organism. Have in mind that's very, very important, and has to do again with complexity, that we tend to think and talk very often about biology in terms of genes, okay? But it's the organism level where most of the things that are relevant happen. And in that context, our organisms work because they are highly cooperative systems, okay? And we don't want Darwinian evolution to be unleashed, something that mutates and replicates and competes. We don't want that. That's what's called cancer or any other proliferative disease. And for that, there's a number of mechanisms of control and regulation that made death an active mechanism. Okay? We are not going to talk about active mechanisms, but have in mind that because of death, which was, I was saying, a major innovation that emerged in the early oceans of our Earth, we can, for example, have a complex development, right? And this mice is the result of a developmental process from a single cell, if you want. Okay? Quite an amazing thing, if you think about it. And in the process of making, for example, the hands and the digits require an active process of cell death, right? That can be seen in the embryo as parts of the bud are removed through the process of development. Okay? So if we go to the population level scale, okay, what is the kind of mathematical model we can build? The simplest mathematical model where no active processes are considered, right? Well, one way of doing that is, again, thinking in an extremely oversimplified picture where, for example, an individual, right, a discrete entity, can transition to its disappearance, right? I use the empty set symbol here, and the idea is that at some rate, an individual can die. Okay? That's completely random phenomenon, okay? But let me say that this is not the only scenario that applies to the model we are going to discuss, okay? For example, if you have a drug, right? A drug that is injected in your organs, in your blood. This drug gets absorbed, and you see, when you measure in blood in terms of concentration, you see how it decays over time because it's being absorbed. You could also say that it disappears, right? Although it's not necessarily a degradation process, it just gets absorbed somewhere else. Or I could have a given molecule, right? We'll see that transitions to another kind of molecule, okay? For whatever reason. Of course, it should be completed with other products, but that could also happen at some rate. I change it into something else. I didn't disappear, strictly speaking, but I did disappear in this configuration. Okay? So all these models apply here. And for example, when you have radioactive decay, you have a given atom that is unstable, and when it loses stability, it transforms into some other element, right? Plus radiation, etc. So how to make a model of that? I'm not going to do all the details because it's very, very similar to what we just saw in the growth part of the story, right? Oops, sorry. Imagine I want to do, again, an equation, a discrete equation, right? Having in mind the same picture, but now this is time, this is my population, right? And my population is going to decay, right? For example, I have cells in a given petri dish, and they are starving, right? They starve because they don't have resources, they die. And what I'm going to see in the most simple scenario is that starting from my initial population, right, I'm going to see how the number of surviving individuals measured just maybe by counting or by some other indirect method quickly decays like this, right? Well, I want the equation for this. And it's easy to build because the process is exactly the same we apply for growth, okay? So essentially is, oops, sorry. I have my previous population anytime, and then I have everything that involves change. Now, change is negative because population is lost, right? How quickly? Well, this is death rate, right? Death rate, in a way, averages any process that has to do with the likelihood that individuals die. Of course, this has to do proportional to the number of individuals. The more individuals I have, the more individuals die, okay? And of course, also, have to take into account my time window, right? Between two given experimental measures, right? Well, the process from here to the equation that defines the mathematical model for decay, for death, is exactly the same, except that I have a different sign, right? It's minus delta x, Okay? This is the simplest equation, linear equation for population decay, right? Either living systems, drugs, or radioactive elements. It says that the speed at which I lose the population, right, because it's a decay process, it's proportional with a constant that says how quickly the process happens to the population that is present, right? From here, it's very simple also to obtain the solution. That's my differential equation for death, and that's my solution. Okay? Which is exactly the same, except for a very fundamental thing, which is the sign. The sign is minus. And that means that now, when I look at the population dynamics, I'm going to see a decay in time. All right? Right? If delta is large, this is going to happen very quickly. Say this is delta 1. Okay? For a smaller delta, let's say delta 2, right? Decay is going to happen with the same kind of shape, but a slower rate. All right? So that essentially is what describes the process of decay, right? And it's a complementary process. When we want to build a model of a population dynamic system, we might necessarily have to consider growth and death simultaneously, right? Except in those cases, as I discussed before, when growth is not limited in any way. So, what is missing here that we need to solve and we need to address in the next lecture? Okay? It's that for the growth process that we considered, right, we make clear the point that there are no limits, and in the real world there are limits. Okay? This is important for a number of reasons. One is that in the real world, when I observe not cells in a petri dish, but a population, I may be more likely to see this, right? Whatever had to happen in the past, it already happened. And I'm observing a population that exhibits some kind of, let's say, equilibrium state, right? And to do that, I'm going to need to understand how things interact when populations are crowded, when resources are limited, okay? And as we will see, when we combine this with the death term, as I just defined, some surprises emerge, right? Some unexpected and counterintuitive lessons can be learned.